All right, so hopefully you can hear me. I'm just working on getting my video loaded here. I believe I have a few people who are already signed into the room, so give me one quick second so that I can do that. Let's see. Alrighty, we have the video started, and I have one person who is currently on the video, and so I have chat up in case you have any questions, and I'm going to give it just 1 minute in case we have anybody else show up who might be showing up right on time. So let's see. Don't see anybody coming in yet. Okay. Okay, so I'm going to go ahead and get started today. You should be able to see my screen, which is the lecture slides for lifespan development, which is chapter 9. So we're going to go through that today. That's going to be, we're probably going to go a little under an hour, which is, you know, about typical class for intro, depending on a day. Okay, so if you don't see my video or my screen with the slides, excuse me, just let me know. But I think right now, I think we're good. Okay, so today we're going to cover chapter 9 with lifespan development, and this is all about what happens from conception to death. And so we'll probably get to about maybe Piaget or so today while talking about different levels of development, different milestones that you hit during development, things like that. So just to give you an idea of what developmental psychology is, anytime we start a new chapter, we've got to go through the terms. We've got to define what it is that we mean by that thing. And so for developmental psychology, this looks at how people change over their lifespan. A developmental psychologist is somebody who's going to look at any element of change that has to do with the stage of development. So developmental psychologists typically cross over multiple fields. So for example, if you're interested in the cognitive factors on development, you might be a cognitive developmental neuroscientist. You might, if you're just interested in behavioral factors, or if you study the brain, you could be a neuroscientist, but also look at a particular part in development. Then that would still make you a developmental psychologist, except that you are multidisciplinary and you can look at many different fields. So this is looking at anything, any influence, biological influence, environmental influence, social, cultural, behavioral influence on development. And if you're looking at a particular stage of life, then that sort of qualifies you as a developmental psychologist. Now, when it comes to looking at the different stages of what happens during the course of conception to death, depending on who you talk to in terms of expert, they may, kind of change this up in terms of different ranges, but generally speaking, this is a relatively agreed upon set of terminology for the different major stages that we see across the lifespan. And so anytime you talk about being in utero, being in the womb, this is the prenatal period, which goes from conception of sperm meets egg all the way to birth. And then infancy and toddlerhood usually goes from that point to 2 years old, early childhood spanning up to 6 years old. Middle childhood is right at the cusp of puberty. So for many, it would probably be about 12 years old, though sometimes we do see children hit puberty a little bit sooner. But then we get through the period of adolescence, which is about 12 years of age to 18. And this varies a little bit culturally, as well as depending on how you define adolescence. Adolescence is usually that period of development where the person is going through puberty. So we do actually have biological markers. Girls change a certain way, boys change a certain way, and we'll cover that maybe in this lecture. If not, the slides definitely cover it. Well, after you go through adolescence, you're moving through this. You're moving into adulthood, but you're not quite, considered an adult in some cultures, especially in the United States. Americans kind of see the early adulthood or emerging adulthood being a period of time where a person is finding themselves. You might still live at home, you might be going to college, whatever it might be. This is referred to more of as emerging adulthood, and that goes to about 25 years of age, and then that's followed by young adulthood, which goes to 40, middle adulthood, which is kind of like middle-aged, and then all the way to late adulthood, which is 65 years to death. So it could be 65 to in your early hundreds, depending on how long you live. We also talk about late adulthood. We, sometimes refer to those individuals as elderly or older adults. There's also acceptable stage names for that period of time. For each of these major stages, you're going to see some different milestones, different transitions, challenges. All these different things are different for each of these stages. And so that's what kind of makes them unique amongst themselves. And of course, within these stages, we have even more milestones and challenges and transitions within each stage. These are just kind of the basic major ones that we talk about. Now, when we look at how do we measure things in developmental psychology, how do we test and assess and research people in different stages, what's really important to know is there are two major kinds of research methods that can give us information about development over time. There's the longitudinal design, where we look at the individual person and we compare them across themselves across a period of time. And then we have the cross-sectional design, which allows us to look at development over time, but not within the same individual. So there's some limitations with that. There's some limitations with longitudinal work as well, and we'll get into some of that. But just to kind of let you know, there are two major methods, but then there are variations of this that you could actually do to either save time or save money. Now, with the longitudinal design, this is a picture of Drew Barrymore when she was a very young child. The reason why I like to use this example is because this is kind of, I think most people know who Drew Barrymore is, and also you can kind of see how this changes over time. So let's say a true longitudinal study looking from childhood all the way through to adulthood. We want to test her at 3 different time points in her life. So with a longitudinal design, it's longitudinal in the sense that it takes a long time to assess them, because you're doing it over a period of time for that individual. So let's say we test Drew Barrymore when she's 4 years old, and that's going to be our time point 1. So she's tested during her childhood, then she's tested again at the second time point during adolescence. So this is another picture of Drew Barrymore. And then finally, we test her again when she's an adult, and this would be time point 3. Now, in a true longitudinal study, this can change in terms of what span are you interested in. Some people are interested in a period of weeks. Some are interested in a period of years. Some are interested in, 20 years. Some are interested in a period of, for example, 4 years. There's a study going on campus right now called the TRACK study, which is following a group or a cohort of students over the course of 4 years. That's a longitudinal study because it's looking at them over a period of time. Now, the good thing about a longitudinal study is that you can make direct comparisons from the same person from when they were a child to when they were an adolescent to when they were an adult. And so the good thing about that is that these are the same participants. Now, you have these measurements over a period of time, but you can directly compare time point 1 to time point 2 to time point 3. And with those direct comparisons, that allows the researcher to make inferences and assumptions and conclusions about how a person changes over time. Now, the downside, of course, to longitudinal studies is that it takes a long time. If this were a real study where we were looking at 5-year-olds, we were looking at 16-year-olds, and we were looking at, I don't know, 30-year-olds over the course of time, you can imagine that, that's a 25-year study. And that takes a long time, 25 years at least. It takes a lot of time to design. It takes time to make sure that you have the right resources and staff to be able to make the longitudinal study actually happen. You have to have people there that will collect data. You need money to do it. You need funding to pay the people, the researchers to do the study, for the staff to do the study. You need money to run the tests. You need usually money to pay the participants. And a downside of doing longitudinal work is that people move, people die, people lose interest. You can ask parents to give consent for their child at 5 years old and 16 years old to take part in a study. But when a person becomes an adult after 18, they can decide, I don't want to do this anymore. So maybe you tested 50 people, 50 children at stages 1, which was 5 years old, and stages 2, which was 16. And then by the time they hit 30, they're like, I don't want to do this anymore. So now you've lost that third comparison into adulthood because the person who has every right to decided they don't want to participate anymore. All research is completely voluntary. So we rely, as researchers, we rely on people to agree to do our research and to volunteer on their own accord. And so, people consent in an informed way that they understand what they're getting into. And the problem a lot of times with longitudinal studies that span childhood into adulthood is that now you have to ask the person if they consent to the study because the parents are no longer the ones who are in charge of them. So there are some downsides to longitudinal studies, but the good part is that it gives us a direct comparison over time on the person themselves. How does that one individual change as time goes on? And then if you test a lot of people over time, then you can see how groups of people change over time. So a way to get around doing a 25-year study or doing a 5-year study and the money and the time that's invested is to do something called a cross-sectional study. And what this is, and some of you may be familiar with some of these characters, this is from the show Full House. So this is Michelle Tanner. She was the littlest one on Full House. And then her sister, oh my goodness, I'm losing her name. It's gone. But the middle sister, and the older sister, DJ. And so I'm so sorry for every middle child out there. I promise I do. Her name will come to me as soon as I log off. So when it comes to comparing with cross-sectional work, the good thing is that you can actually compare participants at the same point in time, but you're making comparisons across the groups. So in this case, we have a person who is in early childhood. We have a person who's in middle childhood, and then we have a person during adolescence. And what you're doing is you're taking each one of these groups and you're comparing them. The one downside is that you can't do quote unquote direct comparisons because they aren't the same person, but you can make comparisons across the board and actually be able to say, this group is different from this group. And so we can make assumptions that this happens in middle childhood, this happens in adolescence, and then make comparisons that way. Now, the downside is that each of these groups are going through early childhood, middle childhood, adolescence at different points in time in history. So different things are going to affect them. You can imagine your childhood would be different from my childhood, and that might actually affect how I am as an adult. It might, affect you differently as you were as an adult. When I grew up, I didn't grow up with a computer in the house. The generations after me, they have computers they have access to computers and the internet even as very young children, and that's going to shape the generations from that point on differently than they shape me. Now, in some cases, that's what the researcher is interested in. That's what the researcher wants to see, is what do these differences look like? Other times they're actually interested in, no, I don't want to know what those differences are. I want to know how similar they are and how similar these different groups are over the different sections of groups. And so in this case, when we're doing cross-sectional work, we have one time point and three different, in this case, three, you can have and more than three, less than three, whatever, three different age groups, and you're comparing, you have a comparison across all three groups. And so this is helpful because it's not as time consuming. It's not as expensive. You don't have to fund a staff for X amount of years. But of course, the downside is that you can't make direct comparisons to the individuals directly because it's not the same person. And of course, historical factors, environmental factors can affect the different groups differently. Okay, so that's pretty much how we go about actually looking at how we research people. Excuse me. Now, when we look at the terminology, so at this point, what we're going to do is we're going to go through the terminology of typical terms used in development. And so if you're a developmental psychologist looking at prenatal, conception to birth sort of that developmental period of time, the terms that you're going to know will be different from somebody who's looking at older adults. But we're going to go through kind of the whole span so that way you're aware of the major terms of each kind of major stage in life. So the first thing we're going to talk about is this idea of genetics, right? So when we're going to talk about conception, we're going to talk about the different terms that we use at different stages during the period of in the womb. But it's important for you to know, just so that, because if I'm going to use these terms, it's important for you to know exactly what they are. And hopefully a lot of these are familiar to you and you've heard them before. You may even know and be able to define them, may know what they are. So a zygote, this is that single cell that's formed when the egg and the sperm unite. And so that zygote has chromosomes from your mother and chromosomes from your father. So the chromosomes from your mother's egg and the chromosomes from your father's sperm, each one of those are going to contribute 23 chromosomes. So you're going to have a set of 23 chromosome pairs. And that set is going to represent your genetic makeup. And so DNA is the really the chemical basis of heredity. That's going to be encoding all of the genetic instructions for every single cell in your body. And so whether your cell is supposed to become a brain cell or neuron or a liver cell or a skin cell, whatever it may be, that set of instructions are going to be in that cell. And that information is going to be determining what cell it is, what it does, how long it lives, where it goes, all those different things. And so one single unit of DNA is referred to as a gene, which is a basic unit of heredity. And you might hear a lot of times people talk about different genes and different kinds of genetic makeups that can change our risk of health. It can change how we look, how we act. It can change traits and things like that. And so another kind of area that I don't have on here when it comes to genetics is something called epigenetics. Epigenetics, epi meaning above, genetics meaning genetics. So epigenetics means above genetics. And I don't really go a lot into detail, and actually I don't even have this on the slide, but I think it is important for you guys to know what epigenetics are. And so what this is looking at how the cellular mechanism works and how that controls gene expression. And so gene expression really does affect your health and behavior. You can have tags on your different genes that essentially what they are on your DNA, and what it does is you can actually affect what tags and get switched on and off. And so you might have a predisposition for something, meaning that your genetic makeup is set in a way that says you have a major risk for developing lung cancer, for example. And so you might have the genetic instructions for lung cancer, but if you don't smoke, eat well, exercise, take care of your body, don't do A, B, C, D, and E, then the right tags won't get switched on for you to actually develop lung cancer. So, and the opposite is true, of course. If you have a predisposition for lung cancer and you do all of the things that switch on the lung cancer, a switch, you might smoke and you might drink and you might, eat poor, eat crap food, and you don't exercise and you do all these different things. And what happens is it sets off a lot of little switches, and that sort of sets a cascade off to eventually change if you develop a particular disease or not. And let's say in this case, you actually develop lung cancer because all the switches came on. And that's how we understand how twins work. So if you have identical twins, and twins are very complicated. People think it's as easy as identical versus fraternal twins. And it's actually much more complicated than that, because you can have twins that are in the same prenatal, that are in the same sac. You can have amniotic sac. You can have twins that are sort of not necessarily sharing the exact same environment in utero. You can have twins that are, that are, and they've just recent studies have shown actually that you can have fraternal twins or almost identical twins that aren't quite identical. They share like a good portion of their DNA. It's wild. It's wild, guys, how twins are so complicated. But if we look at, and we try to break it down to basic, if we look at a set of identical twins, they share 100% of their DNA. They are essentially one egg and one sperm that united, right? And they split. It's one egg that, or excuse me, one cell that splits into two, and these two create two people. Now, with fraternal twins, you have two eggs. So a woman releases during her cycle, she releases two eggs. And so one sperm gets this egg and one sperm gets this egg. And so what happens is you have two sets of DNA that's different. It's like brother and sister, or sisters or brothers that are born at different times. It's different sets of DNA. And so these just so happen to be in the same place at the same time. And so they grow up in the womb together and become twins, right? Well, in identical twins, we have one egg and sperm unite, and then that is one set of DNA, and then it ends up splitting into two people. And so what ends up happening is we have an identical set of DNA. We have two people with identical set of DNA. And so what these studies were done to look at, how is it that you can have two people that share the same exact DNA, but yet over time, one person might develop breast cancer and the other person doesn't. Or one person might get heart disease, but the other doesn't. If it's all genetics and everybody's in the twins' DNA are the same, then how do you explain the genetic predisposition for something? If you have a genetic predisposition, that means you should definitely get it, right? Well, it turns out that's not the case. And so that's what taught us about epigenetics, is that is malleable. There's still environmental influences that can impact whether or not you actually develop something like breast cancer, lung cancer, heart disease, obesity, all these different things. And so what ends up happening is that we have these two separate people, even though they share DNA. This person, doesn't exercise. They have a very stressful life. They smoke, they drink, they have an alcohol problem. They have all these environmental things that they're doing wrong, that switch on all these different things. And maybe that person develops liver cancer. Then you have this person over here, and they have the same foundation of genetics. This person over here has a genetic predisposition for liver cancer, but they eat well, they exercise, they take care of themselves. They're not super stressed out all the time. They really pay attention to their body and their mind and everything. And this person doesn't develop liver cancer because those particular genes aren't switched on. or those particular tags or alleles aren't switched on. And so what ends up happening is we realize that there's a lot more to genetics and epigenetics and, what makes you who you are. There's so much more than just, cells and genes and DNA. So this is a really interesting kind of way to look at development, because development is very much impacted by your genetics, but it's also very much impacted by environment. And so when we look at what's more important, genes or environment, for a long time, psychologists especially would argue one or the other. They would say it doesn't matter what your biology says, it matters what your environment is. So people would say that you are a blank slate. So psychologists used to use that term blank slate. So it doesn't matter if I take one person and plop them into this really well-to-do home with a lot of resources, that person is going to be an excellent contributor to society. If we make them eat healthy food and exercise, that person's definitely going to be healthy and live to an old age. Well, I'm sure you know plenty of examples of people, friends that you know, who have, rich parents. They have nothing to worry about, but then they engage in bad behaviors, or they, die early, or they get sick, or there's all sorts of things. So it turns out that we're not blank slates. It's not all about environment. You can take a person and put them in a really bad situation, and that person might crawl their way out and be, president, or be, a doctor or a lawyer, and be, amazing. And contribute to society, even though they had a really, really poor upbringing or a bad upbringing, or, whatever. And so on the flip side of that, right, genetics aren't everything. Genetics are a part of it. So now we know that it's a mix of genes and environment. So if, there's this kind of, argument, is it nature or is it nurture? And for, a long time, psychologists would say it's either nature or either nurture, but now we know it's both. It's both nature and nurture. It's your environment and your genetics, and that determines who you are. And it's the mixture, and they interplay back and forth. So environment is going to influence your genetics, and your genetics is going to influence your environment. And it's just a complete, integrated married relationship. Now, let's talk about some terms in prenatal development. Okay. So here we talked about this idea of conception. This is where the sperm is going to meet the egg, and this is where the cells begin to start doing stuff. So conception is when the egg and the sperm meet, and that forms a fertilized egg. So once the egg is officially fertilized, it's called a zygote, and then the zygote is going to start changing very, very, very quickly. And during the period of the first 2 weeks after conception, we refer to this as the germinal period. And then when that's over, up to week 8, that's considered the embryonic period. And then after that, 2 months after conception, all the way till the fetus is born, is considered the fetal period. So a lot of people get really upset when they hear somebody say fetus over a baby. And really what it is just using proper terminology. In developmental psychology and in the developmental world, fetus or fetal period refers to the period of really 8 weeks to birth. So if, you're using the term fetus and the fetus is in the womb, then you're using the correct terminology. It doesn't become a baby until it's born, because that is the correct and agreed upon terminology in the developmental field. Now, during that period of embryonic period, so I want to remind you, we're going to go back. The embryonic period is weeks 3 through 8. It's a very critical period that's right before we hit the fetal period. This is an especially vulnerable time for the embryo. Okay, this is a very vulnerable time. This is where a massive amount of formation happens in the organs, in the body, in the brain, in the cells. The cells are moving. They're building the layers that are going to make up the brain. And so what happens is if you as a pregnant woman are exposed to something that's harmful, and we call these teratogens, a harmful agent, a harmful substance, during this period of time, there is an increased risk of experiencing of that at the time embryo and then later fetus becoming malformed or defective. And so you have to be really careful during this time because it's especially vulnerable. This is when the cells are forming and the brain is forming really from scratch, right? So anytime you're exposed to radiation. So for example, if you go to the dentist, a lot of times dentists will, I think every dentist should have this, a paper listed up in the front that says, if you're pregnant, please let your dentist know, or please let your hygienist know, or whatever. Let us know that you're pregnant. And that's because if you needed to have x-rays done of your mouth, they'll actually usually, unless it's like a serious issue where, you know, it's like a health issue. If you're just getting routine x-rays, they'll actually wait and do it. If you suspect that you're pregnant, they'll do it later. If not, that there's ways that they can kind of cover you. But the idea, and they'll use like an iron, you know, apron. The idea is that if you are exposed to radiation, radiation actually affects and mutates cell formation. So the cells will mutate and it could cause all sorts of issues for the embryo or fetus, depending on what stage you're at when you're pregnant. Any kind of toxic chemicals, metals, you know, lead paint, anything like that can cause issues with the embryo or fetus. Any kind of viruses or bacteria can kill the embryo or fetus. It can also cause a lot of issues. And when we get to abnormal psychology, which is the last chapter in the class, we're going to talk about the fact that there's actually a link between viruses in utero. So a woman who's pregnant gets sick with a particular virus, and that gets passed through the bloodstream to the fetus. That can actually put you at risk for developing schizophrenia. And so what we found is that there are certain viruses that turn on and off certain genes and certain things, and that can put you at risk for disease. There's a theory that Alzheimer's is caused by a virus. And so it starts early on when you're younger, and then that virus turns certain things on. And then over time, you'll actually start developing, over a very long period of time, slowly start having changes in your brain. That's one theory of what causes Alzheimer's. So any kind of viruses, bacteria can cause major issues. You know, it can cause any kind of condition or sexually transmitted infections, sexually transmitted disease. Any kind of virus, bacteria can cause things like blindness, deafness. It can, you know, really, really harm the embryo or fetus. Anytime you take either prescription drugs or non-prescription drugs or illicit drugs, that can have an effect on the fetus or embryo. And the idea is that if you are a pregnant woman and you are prescribed a certain medication and you need that medication to live and you need that medication to survive, then usually your doctor will look at you and say, we need to weigh the benefit versus the risk. And if the risk is you're going to die or you're going to be harmed, or that's, you know, a problem for your health, it's going to make you unhealthy, then usually the doctor will say, we will continue the medication or we'll change you to something that we think is safer. Any kind of illicit drugs, any addictive drugs, smoking, secondhand smoke, all these things can impact the developing fetus. And in fact, we know that babies who are born to mothers who either smoked while they were pregnant or there was somebody who smoked in the home, so they were exposed to secondhand smoke. Those babies are actually born smaller, so they're born, they weigh less, and a lot of times they go on to develop asthma or they have issues with lung development. So this is something that's really seriously important, that if a woman is pregnant, she really needs to make sure that she has a very healthy environment. Not only do you eat, you know, better, you eat good, and you eat, you know, well, and then you take care of yourself, make sure you're not taking any illicit drugs or addictive drugs or anything like that, but also that you have a healthy environment. Alcohol is a big one. Now it's really it's kind of sad when you think about it, but what happens with alcohol is that the baby could, or the fetus or embryo could be affected in such a way that alcohol can cause fetal alcohol syndrome. And fetal alcohol syndrome is one of those absolutely 100% avoidable conditions. All you have to do is not drink during pregnancy. And that's how you avoid fetal alcohol syndrome. And you would think to educate people, say, okay, if you drink during pregnancy, that's a bad thing. You would think the education works, but it turns out it doesn't, because the rates of fetal alcohol syndrome over the past 20 years actually hasn't declined much at all. And so telling people not to drink during pregnancy isn't working. Many people know that it's unhealthy to drink during pregnancy, but still engage in that behavior. And so we've looked at, there's rat studies and mice studies that look at when is it safe to drink during pregnancy? And most scientists will say it's never safe to drink during pregnancy. But if you had to look at binge drinking, so drinking a bunch, like taking a whole weekend and just binge drinking the whole weekend versus drinking like, I don't know, half a glass of wine every night, binge drinking tends to be actually more harmful than consistent, very low amounts of alcohol. And this is also cultural. So, you could look at, for example, France, who they consume a lot of wine during meals and things like that. There's actually less of a stigma on consuming small amounts of wine during your meal if you're pregnant, compared to in the United States, which usually if you see a pregnant woman drinking wine, somebody's going to say something to her is what I'm getting at. So there's a cultural difference here too. And some researchers say that once you hit a certain period of development, or once you hit a certain number of weeks pregnancy, that the risk and the damage is less. Yes, there are definitely critical periods of time of development during the development of the limbs and the organs and things like that. But really, if you want to make sure that you're not going to harm your fetus or embryo, you just want to avoid it. Now, another thing that's really interesting when it comes to like prescription drugs, there was a medication that was used. It was a prescribed medication. I believe it was used in the 60s, and it was for morning sickness. So, during pregnancy, you can feel nauseated. And so there was this particular drug that was designed to help women not feel as nauseated in the morning. And so it was given to women during the early stages of pregnancy. And it turns out that when it's given during a certain amount of weeks, there's a very small window, when it's given during the window where the embryo and later fetus is developing limbs like arms and legs, if you take the drug during this period of time, remember, this is a prescribed drug from a doctor, it will actually result in the child not having limbs. It stunts the limb growth. So the child is born with either stumps or no limbs at all. And so several babies were born without legs and arms. And everyone was like, oh my God, what's going on? And it turns out that this particular medication was okay when it was given outside this range, and it would help the mother not feel sick. But if it was given during this particular window, it actually caused devastating effects on the fetus. And so we know now that certain drugs given certain times can have different effects on the embryo or fetus, depending on what that drug does. So just because a drug looks fine and is tested and appears to be something that's helpful, it doesn't mean that it's going to be good during pregnancy. So again, the risks and benefits have to be weighed when you look at somebody who needs a medication during pregnancy. And then also, like I was saying about, creating a really good environment, really healthy environment. If a woman is completely stressed out and is in a poor psychological state or engages in really unhealthy behaviors, that will translate to the embryo or fetus. If you're going through a major trauma, if you have a lot of issues with, being in an unstable environment, if you're homeless, all of these stressors can impact the embryo or fetus. And so that can be majorly problematic. One thing I do want to go back, I didn't say this specifically about fetal alcohol syndrome. I don't think I explained it well enough in terms of what it is. So when you consume alcohol during pregnancy, and enough for the fetus to develop fetal alcohol syndrome, the kinds of symptoms and effects that you see, it actually changes the shape of the face a bit. And so you'll see things like a very smooth upper lip. The eyes are a little bit spaced far apart. There's some interesting abnormal facial features that happen. We also see that many, and this is a spectrum. So you can have somebody who has a very mild case and they turn out to be very functional, children and adults. And then you can see very severe cases where the person is completely mentally handicapped and is not able to function at all. But so there's a range here, but we see things like issues with coordination, their balance is off and they're very clumsy, learning disabilities where they have a difficult time focusing. We see a lot of behavioral issues, especially during the school year period, where they act up in class and they have a really poor impulse control and can't control themselves. And then of course we see just intellectual disability. So the child is mentally handicapped. And so these are all things that are completely avoidable, like I said, if the mother avoids alcohol. Of course, we can always look at cases where a person didn't know they were pregnant and engage in consuming alcohol. And obviously once the woman knows that she's pregnant, of course, she should not engage in that behavior anymore. So let's talk a little bit about what happens prenatally, okay? So this is a really interesting sort of the way that this works. So this is 3 weeks here in utero. And I'm going to get a pointer. Actually, I'm going to do annotation. So I'm going to draw on here. Hopefully you guys can see this okay. All right. Yes. Okay. So I should be able to draw a line here. Let me test it. Yes. Excuse me. Okay. So during this period of A right here, this is 3 weeks in utero. And as you can see, it already has some form of something going on there. Now, a lot of times if you look at different animals in utero and different stages of development, they all kind of look alike. So you all kind of look like a little bit like a shrimp, but then eventually you'll get many more features. So during this period of development, the cells are moving to their locations. They're all stem cells and they can be whatever they want. And they can be brain cells, they can be skin cells, they can be whatever. And so before it becomes a neural tube, okay, what we have is something called the neural plate. And this is a very crude drawing. I have my mouse pad, not my mouse, just the pad on my laptop. So I'm going to try to draw this as best I can. So just bear with me. So this right here would be the neural plate. Okay. Ooh, this is terrible, but you guys have to deal with it. Sorry. Okay. So this is the neural plate, and this is what starts before it becomes a tube. It is a plate. And so the plate then develops, and again, this is really crude, something called a groove. Let's see if I can write groove. Okay. And so it goes from a plate to a groove in a very short period of time. And then it kind of closes in on itself to form the tube. And then that is the precursor of the brain. If you take human neuropsych or biopsychology, you're going to learn a whole lot more about this, and this is really fun. Okay, so here we go. So it starts with this plate, moves into a groove, and then those cells kind of collapse upon themselves to finish a tube. And then the tube becomes, so it starts, this brain begins this as a tube right here, right? So you can have cases like spina bifida, you might have heard of this, is when the tube doesn't close properly. And so spina bifida can be where the tube doesn't close properly and you have major issues with the spinal cord. And so there was, in the 80s, it was really big to get tested to see your risk or the embryo or fetus's risk for spina bifida and see if the tube actually closed, because they were saying, doctors would say, okay, you know, we have to look at how severe this might be. You might have a fetus that isn't going to survive because the tube didn't close properly and now the spinal cord is a mess. Or it might be as mild as, you know, the person can have surgery and get it fixed, and you don't really see that it has a major issue. So that can range in severity. So this right here, so it goes from a plate to a groove to a tube, closes in, and then starts to, and is filled with fluid, and it starts to bulge out at different areas of, to make up the different parts of the brain. So this tube is lined with stem cells, which navigate themselves to different locations on the tube to become different parts of the brain. So here we have, in this case, this is the tube bulging out and thickening into different areas. This is the forebrain, that's pink. The hindbrain is green, the midbrain is green, and the hindbrain is blue. So from 3 weeks to 11 weeks, we see what looks like a really smooth brain. Now, when a brain is forming, right, it's gonna be smooth. But as the cells start to increase, it's the surface area, it's got to go somewhere. So it kind of like folds in on itself. And so imagine taking a piece of paper. Let me grab a piece of paper, something that I'm not using here. Am I using this? I'm not using this. Okay, so let's say we have a piece of paper here. There's a lot of surface area here, but it takes up a lot of space, okay? So imagine this in my head. That's a lot, right? It's too much. Now, let's take this and let's make it smaller, right? So now you've got all these little crevices, right? All these little wrinkles and crevices so that you can fit this into, in this case, the palm of my hand. Now, originally, that sheet of paper is not going to fit in there. Think of this as the brain. The brain is going to keep folding in on itself. The cells are going to keep multiplying and moving, right? So that it can fit in your skull. Because if you have just a smooth brain, that's a lot of cells, but you can make a lot more cells. And if you make more cells and shrink them up into wrinkles, you can actually fit more surface area and more neurons into a smaller capacity, which is your skull. Now, here we can see that at this point, this forebrain here, which was pink, has taken over. It's bulged and taken over a big portion of the brain. And what this becomes is your cerebral cortex. It becomes all the cortex outside of your brain. And so here your forebrain starts to get bigger by 11 weeks. Then the midbrain is still visible. You can still see it. The midbrain in green here, you can see is still behind the forebrain. And then you can see the midbrain starts squishing up, okay, and it's starting to become the cerebellum and the base of the spinal cord. And again, this is visible and it's not quite formed just yet. By the time we get at birth, the cells have multiplied. They have the synapses and everything have formed and the connections are meeting up and all things are squished together. The midbrain now, as you can see, is not visible. That is in here. Now, I'd have to hack this off in order to be able to see the midbrain now. That is housed into this outer portion of the cerebral cortex. And then, as you can see here, the midbrain has squished up and turned into the hindbrain, or in this case, you can see very clearly the cerebellum. And then now you can see the base of the spinal cord here. So these neural stem cells are dividing, they're multiplying, they're moving, they're transporting along this tube to form these different categories of brain matter, right? They produce neurons, they produce glial cells, and glial, remember, means glue. This is the glue that holds all the cells together and all the in-betweens, right? And all of this works and forms in just a short period of time. We're talking 40 weeks from beginning to end. We go from, in this case, 3 weeks, right, to birth, to 40 weeks, so 3 weeks to 40 weeks. And this is an insane amount of development in a really short period of time. So let's see here. Now let's move to birth. Okay, so now we've looked at brain development and we've seen how that works. Oops, let's go ahead and clear this off. Now, when you get to birth, okay, when you're born with several different reflexes. Now, if you look down here, there's a YouTube video, and this is a really fun video. If you love babies, you're going to love this video. And so this is kind of like the a visual of the kinds of reflexes I'm about to talk about. So rooting is a basic instinct for the baby to know how to survive. Okay. So rooting is when if you brush and let me just warn you, okay, if you know somebody with a newborn baby and you want to try this out, clear this with them first, just because it's always a good idea. Don't say, oh, my professor said you should totally try this on your baby, and she's like, telling me to stick my finger in her mouth or whatever. Please, I didn't say that. I'm going to deny it. I don't care. It's recorded. If you want to try this out on a baby, always ask somebody first, right? If it's your baby, then, ask yourself for permission. But anyway, so rooting is if you brush the baby's cheek, okay? What the baby's going to do is going to root for the source of that, okay? So it's going to turn its head to the source of the sensation. And rooting comes from this idea that the baby is looking for a nipple, right? So the baby is rooting for a food source. And so whether it's a nipple on a person's breast or it's a nipple on a bottle, it doesn't matter. Instinctually, the baby is looking for a nipple on a breast. If it's brushed up against, it's going to go to that, and then it'll suck whatever it comes in contact with, which is another reflex. So sucking is this rhythmic response to oral stimulation. It's a very intricate coordination of breathing, swallowing, and sucking. And not all babies master this directly at birth. If you have a premature baby, they might not quite have the resources to suck yet, and they actually need time to develop this. And newborn babies, though, who are at term and they're developmentally normal and fine, what we do find is many times they have a perfectly fine suck reflex, and they get it down pretty quickly, just literally directly after birth. And in some cases, you'll pull a baby out like of the womb and they're already sucking. And so that's a really interesting reflex that is obviously important for survival, because if it wasn't an instinct, then the baby couldn't eat. Now, Babinski is one that we're not quite 100% sure why we have this reflex. So if you have a baby's foot and you stroke across their foot, what happens is their toes will fan out. It could be something to do with a precursor to walking or balance or something like that. We're not 100% sure why, but it turns out you lose this reflex after you're an infant. And so if you're an adult and the Babinski reflex comes back, that can actually be a sign of a neural issue. And so that's something that actually is tested even in adults to see if they have the Babinski reflex, because it could be indicative of a problem. And then grasping is if you take your hand, your finger, and you put it in the palm or near a baby's hand, they will grasp your finger so tightly that you can actually lift the baby up. Now, this is a scary one because obviously you don't want to drop the baby, and the baby can only hold its weight for just a very short period of time. But this is a natural reflex. And so this goes away as the baby starts to be able to control their hands, and no longer is a reflex and then becomes a voluntary thing. So at first, when the baby's very young, very, very, very small infant, it is a thing where it is just a natural thing that they do. But over time, they'll be able to gain control of this, and it no longer is a reflex. Now, what's really interesting about infancy is that brain of theirs grows really considerably. It grows to about 75% of its adult weight during infancy. That's a lot of development that happens in that brain during a really short period of time. And we also see two patterns here. So we see what we call a cephalocaudal pattern, which is what we call kind of top to bottom. So this would be where we see that improvement happens in structure and in function, first in the head region. So if you look at a baby and you hold a baby, the first thing that the mom's going to yell at you is support the head, support the neck. Because if you take a baby that's brand new born and you pick it up, the head's going to flop over. And that's because they don't have the neck strength to keep their head up. So the first thing that a baby can accomplish really is to kind of keep their head straight up because those muscles have to form. And so this is why babies kind of look like little turtles, is because, you know, they're gaining that strength in their neck. Then they can hold their heads up. Well, then their trunk strength happens where they can start to sit up, and then later it goes down to their feet where they can walk. So think of it like a head to foot development. It's a pattern of development where we see that it starts with the head and the neck region and moves all the way down to the feet. And so the trunk and the legs are the last place to develop. Now, proximal distal refers to a pattern of development from center onwards out to extremities. So here you can think about how growth starts at the center, and your ability to control your body really starts at your trunk and moves its way out. One of the last things kids are able to do, if you think about development, they can, you know, kids learn to sit up, they learn to crawl, they learn to walk, they learn to do these things, right? But it's not until they're like 6 that they start to be able to control their fingers really well, right? So it goes from in and then works its way out to the extremities. Fine motor movement is one of the last things that we see children being able to really get a grasp of. So, I mean, you know, you can't hand a 1.5-year-old a crayon and expect that baby to be able to draw a letter or write its name, right? You know, you see the kids, even at 6 and 7 years old, will sometimes hold a crayon, like kind of like a caveman, and they're like, you know, coloring. As the child starts to age, you're going to see that they're going to be able to start holding things in a way that's controlled fine motor movement, and they can actually write and they can actually control that. So proximal distal just simply means that, you know, the extremities are the last to really form in terms of development. Now, when we look at development across the board, universal, across all people, it doesn't matter where you're born, doesn't matter when you were born, doesn't matter if you were born in, you know, Africa, or, you know, a country in Africa, it doesn't matter if you're born in Kenya, it doesn't matter if you were born in New Jersey. The basic sequence of motor skills is completely universal. And this is a human thing. It's not a cultural thing. So the averages are all there, right? The basic sequence is all there. A baby is not going to be able to walk until it can hold its head up, right? So the sequence is there. You know, being able to have the strength to hold the head up, being able to, you know, sit up, being able to stand before it can walk, to, you know, to be able to crawl, to have the ability to crawl. And some babies just choose not to crawl, have the ability to crawl before the baby can run, right? So even though this development is universal, we can see the averages can be a little bit deceiving because it's a range. It's an average of a range. So here we can see rolling over, the average age is about 3 months, right? But if you look at this, you can see a baby rolling over as early as 2 months or as late as 5 months. Same thing when it comes to walking. Babies can walk as early as 11 months and even sooner than that, all the way down to almost 15 months. And so people, especially parents, get really panicked because they're like, my child isn't doing this yet. Oh my goodness, what's going on? And it turns out that a lot of times it's just a matter of that your child is normal and everything is normal. It's just a matter of this is not, that they are just developing at a slightly slower rate than, you know, it's still considered normal, but slightly slower compared to another baby. And so, you know, you have to kind of look at this as a range and know that, yes, there does come a point when development is too slow and there might be something wrong. But usually children are fitting in to a range that's completely normal, and parents sometimes freak out. Now, the last thing I'm going to talk about, I think, in this lecture, unfortunately, I really wanted to get to language, but we're not going to get to that today. The last thing I want to talk about is the forms of attachment. And the reason why I want to talk about this is because this is something that is hard to get from the slide, and I want to make sure that you guys understand what happens here. So, and then I might say one thing about language. I might go over just a slight bit, but if you got to leave, I understand. This is going to be recorded anyway, so you can look at it later. Now, attachment is this idea that attachment is really, really important, because this is the emotional bond that forms between your caregiver. Doesn't necessarily have to be your mother or your father. It's whoever's taking care of you, your caregiver and the child. And so Mary Ainsworth did an amazing set of experiments where she used the strange situation procedure. And so what she was interested in, and at the time, this research was done several decades ago, but at the time she was mostly interested in the mother's role. But now we've learned that the father's role is also very important in this idea of healthy attachment. So when we look at the strange situation procedure, essentially what happens is you're interested in how the baby acts at each point. So at the beginning, you have the mother, and this, I'm just going to say mother because it's easier, and this is the original experiment. You have the mother walk into a room with the child, and then she sits in the room, and there's a two-way mirror or a camera, and so people can actually watch this as it's happening. So what happens is the mother is sitting with the child, and the researchers are interested in, what is the child doing? Is the child exploring? Is the child interested in the environment? Or is the child just simply looking at the mother and too scared to move or whatever? Sorry, I'm just making sure my laptop isn't overheating. So you're looking at the behavior of the child. Then the mother gets up and leaves the room. And so you're looking at the behavior of the child. Does the child get upset? Is it exploring the room? Does it stop? Does it get mad? What's going on? What happens during that time? And then there's variations of this. One could be a stranger comes into the room, okay? And they want to see how the child acts without the mother. And mostly, though, the traditional experiment is the mother comes back into the room, and the child is observed to see, what does the kid do? Is the kid excited to see the mom? Is the child upset to see the mom? What happens? And so and this is a big area. This isn't just, I've simplified it to one slide, but you could do a whole class on this very easily, because this is a lot of work involved in this. So basically speaking, a secured attached infant would be one that, when the mother is in the room, feels safe enough, even in a strange room it's never been in, to explore and look around. And then when the mother leaves the room, the child becomes upset because the mother has left and is not going to continue exploring the room. But then when the mom returns, the baby's like, "Mama," and is excited to see the mom. And so the child's not upset at that point, is happy, and everything's fine. Now, an insecurely attached baby. Now, this is one, there's variations of this. This isn't just insecurely attached, isn't just the label. There's different variations among an insecurely attached baby. Would react very differently. They may not explore the environment initially, even with the mother. They may not do anything when the mother leaves. They may not get upset. And some babies will get super upset when the mother returns, but they're inconsolable. So the mom might be like, "Oh, it's okay, baby," but the baby might be fighting her, might be slapping at her, might be angry. The baby might completely ignore the fact that she's gone and then comes back. There's a lot of different variations of behavior, but ultimately the baby does not seem happy to see the mom, or is angry at the mom, or is indifferent to the mom. And this is a sign of an insecurely attached baby. And again, there's a lot more into this. So if you're interested in it, there's certainly a lot of work out there to read. And so the idea is that attachment's very important, because it's the beginning. In infancy, this idea of emotional bonds, it forms the basis of emotional bonds through childhood, through adulthood. So the idea is that if you are insecurely attached as a baby, as an infant, to your caregiver, this could mean that later on down the road, you might have difficulties forming attachments to others, like your spouse or your partner or a loved one or whatever. Or you either might have difficulties with this, it might not come as easy, or it's abnormal. So you might have insecure attachments to romantic partners as adults. Now, it's not completely damning. Just because you had an insecure attachment as a child doesn't mean, or an infant, doesn't mean you're going to grow up to be somebody who's never going to be able to find somebody who's not going to be in a good relationship and a healthy relationship. That's definitely not the case. Even though it does put you at risk, there are other people in your life that can bond with you at an early age that can save you. And also, even if you were in a situation where you didn't have a good attachment to anybody, if you're exposed to really healthy relationships in childhood or early adulthood, that can actually set you up to have a really healthy relationship as you age. So even though, it kind of is a crappy a beginning for some people, it doesn't necessarily mean they're going to end up not being able to be in healthy relationships. All right, the final thing I want to talk about is language development. And this is because it's kind of fun, but also there's a couple things on the slide that I want to make sure that you guys understand. And then the next slide is about Piaget. And so I have got a table and a video, and I think it's a really good explanation of Piaget's stages and what happens in each stage. So I think you guys will be good beyond this point. So let's just talk real quick about language development, and then I'll wrap up and then we'll be done. So Noam Chomsky is a really, really famous linguist who talks about language development and says basically that everyone has the ability, every human born has the predisposition to learn language. Any language. It doesn't matter. It doesn't matter what language that is. Anybody can be taught any human language when they're an infant, and then up to, as they grow up. Now, so there are so the idea is that everyone has the capability of understanding and learning language. So there is a critical period during development where you actually will lose the ability after about a year old of being able to identify any sound and replicate any sound between a specific language. So up until about 10 to 12 months of age, during the birth to 10 months-ish, you as an infant can distinguish the sounds of any language. Doesn't matter if it's a click language, Germanic language, doesn't matter the language. You can distinguish any sounds of any language. If you do not listen to or hear the language after you turn a year, you lose this ability to learn a particular language, or lose the ability to learn a language outside of your own in a way that is like a native speaker. Okay. Now, can you learn languages when you're older? Absolutely. Can you learn fluently? Absolutely. Can you sound like a native? Sure. But this is an ability that we have, a critical period that we have, that is the easiest point in development to learn language. Now, by the time a child reaches about 3 years old, it turns out that they have about 3,000 words that they've learned, and a lot of complex rules of language. So our ability to understand language is insane, and it's also very quick, and it forms very quickly, and we are very capable of learning language. There are some universal characteristics when it comes to language development and what we see in languages across the world. Universal. Every human, doesn't matter the language. So one thing is called infant-directed speech, or motherese, or parentese, which is a style that we see very distinct pronunciations. We see that it's simplified and shortened. We see that in any time that you think about how you would talk to a dog, oh, how are you doing? Oh, this is such a sweet little baby. You're just so cute. Okay, that is infant-directed speech. It's high-pitched. It's exaggerated. You're opening your mouth a lot because you're like, oh my goodness, you're such a sweet baby. That is super annoying. Sorry. That is infant-directed speech. You're going to restrict content that you think the child's going to understand and is going to know. Children love it. Babies love, they respond to it. If you have an infant that's 4 months old and you're like, who's this cute little baby? The baby might actually mimic you and is going to react to that. And so that's why we instinctively use it, is because infants prefer it. And so that increases the likelihood that we're going to continue using it, because when you do that, you see the baby might smile or engage with you, or at least pay attention to you. And so we see that that we'll do things like, Do you want to, let's go night night. Okay, mama go bye bye. These are all simplified vocabulary. We do shortened sentences so that the infant or the baby or the child can follow along. We want to make sure that everything that we do is a way to set up the infant or the child or the baby to be able to learn the language. And one final thing that I really want to make sure you guys know. Now, one of the first things that a baby is going to do in terms of language, where they're actually making noises, is coo and babble. Those are the first things. So like, ah, blah, 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 you know, and so when I was watching one of my nephews one time, he was 6 months old. And so cooing actually happens around 3 months of age, where we see like, ah, ooh, ooh, right? But my nephew was a little bit older and he was babbling. And we see this start around 5 months of age with consonants and vowel sounds. But at 6 months-ish, I remember one time I was feeding him in his high chair, and he said to me in his 6-month babble, and he was mimicking language. You know, he didn't make any sense. And so I was like, what? What are you talking about? And no lie, he looked at me like I was the dumbest person in the world. He looked me straight in the eyes and he went, and he repeated exactly what he said. Basically, he was like, look, idiot, I just said this. How can you not understand me? And so to babies and to infants, their babble is language. They are engaging with you. They're talking to you. To them, they sound like they're speaking. And so this is the first precursor to language. Cooing, babbling, cooing, you know, these are the first sounds that they get to understand how to move their mouth. And what's really fun is, in terms of babbling, even deaf infants who do not speak babble in sign language. And it's really neat. It's something that we all do. And we see that over the course, after, you know, the 3 months cooing, and then the 5 months of babbling, and then the 9 months of you know, babbling that's more specific to their language, we see that this comprehension starts to increase and start to develop even more. And this is the precursor to giving us the ability to be able to make the language sounds that are specific to our language to allow us to communicate to one another. Now, the next set of information is all about Piaget. And like I said, you've got a video and a table. I've got my chat up. I don't have any questions or any comments or anything, so I guess that we're pretty good on that aspect. If you're watching this as a recorded lecture and you have any questions, you know, you can email me. Make sure you you know, look at your quizzes and look at Moodle to see all your assignments that are due, because everything is up on that. And if we don't have any questions, I'm sorry I went over a little bit, but I really wanted to get to that because I think that's really important to know, and that's not necessarily, some of that stuff is not necessarily on the slides. So if you have any questions, let me know. If not, hope you guys have a great night, and we will see you next time.